So we've gone through all of the main electrophysiological methods. We've seen their uses. Let's just go in one more step of detail, right? Which is to say, if you had to read a paper with these techniques, what would you need to be thinking about? So all extracellular recordings, if you're actually looking at the waveforms, can be a bit tricky to figure out, right? So this, what we see here in the middle in C, is the most classical example of the field excitatory postsynaptic potential. So field, meaning it's recorded outside of the cell. It's a field potential. It's a local field potential. It's the voltage outside the cell. And it's a field excitatory postsynaptic potential. So this is what you see outside the cell when a whole bunch of cells are being depolarized. And the best place to do this is in CA1 of the hippocampus. So what you end up typically doing is stimulating these axons that are called the Schaefer collaterals. They release glutamate onto these CA1 pyramidal cells, and by stimulating them, you cause all of the Schaefer collaterals to be active at once. They release a hell of a lot of glutamate and depolarize a hell of a lot of CA1 cells, and all of that sodium rushing in through all of those AMPA channels causes a significant field potential, which is what we see here, right? So the first little straight up and down line is probably just the what's called the stimulus artifact. That is the fact that you're stimulating the electrode, zap the tissue, and you're able to record that. But then the next bit, this is the local field potential. Now, it goes down the page. Why does it go down the page? Because when you have your electrode in amongst the dendrites, you're seeing sodium go into the cell through those AMPA channels, and a positive sodium going in makes the extracellular voltage negative because, there's slightly not enough positive charges anymore. And so then when you apply CNQX, which blocks AMPA channels, you can see that there's very little of the EPSP left, but these stimulus artifacts are just the same. Now, this is a simulation that I ran of the idea that these local field potentials are confusing, right? So if you stick your electrode down here in the dendrites where the Schaefer collaterals are, you see this negative going potential. Like I just said, the sodium goes in, so that makes the outside negative. But as I said way back at the beginning, charge cannot be created or destroyed. So if sodium goes in, then a positive charge needs to leak out somewhere else. And so if you put your electrode up by the soma, or possibly even in these dendrites, then you see that effect of the positive charge coming out. And so now the local field potential goes in the other direction. Even though if you were inside the cell, in both places, you have a depolarization. Now, this is like, I can clearly remember being deeply confused by this, right? And thankfully, this isn't a biophysics course, so we don't need to get into a lot of details. I'm using this to highlight a couple of things. Firstly, local field potentials are easy to get confused by. But if you have your electrode where the synaptic behavior is happening or the ion is flowing into the cell, it's easy to interpret, relatively speaking, right? Just like I said, we see sodium going into the cell, so the positive goes away, and that makes the extracellular environment more negative, and so you see a negative going potential. This is a real recording where they've moved the electrode further and further up, and you see just what I demonstrate here, that all in and amongst the dendrites, we have this negative going wave, but as soon as we get near to the soma or up into the other side of the dendrites, then we see a positive going wave. Now, if you stimulate a cell hard enough, of course, these EPSPs cross threshold, and that causes an action potential. And so you can see that extracellularly as well. So if you put your electrode up near the soma, or really near the axon, if you stimulate hard enough and get a big enough EPSP, you get an action potential. And what happens during an action potential? Sodium goes into the axon through voltage-gated sodium channels, and that's exactly what we see, or most clear here, right? So you see a negative going potential as the sodium goes into the axon, but it's sitting on this positive going hump. Why is that? Because, of course, as I said before, during the EPSP, sodium goes in at the dendrites and then positive charges come out at the soma and at the other dendrites. So you see a positive going hump, which represents this charge coming out of the cell, and then you see a negative going spike, which is the action potential. Of course, it's worth remembering that we can only record these so clearly outside the cell because there are thousands of cells doing the exact same thing. And they're all packed together in the hippocampus, and so it creates a very clear extracellular waveform. Now, this experiment, this kind of approach was famously used by two scientists called Bliss and Lomo when they discovered the idea of LTP, long-term potentiation. And here you can see their field potentials, and then they applied a tetanus, which is like a high-frequency synaptic activity, and you can see that the spike got bigger. Now, this may be confusing at first. Action potentials are all or none. This population spike, this extracellular spike, is caused by action potentials. So how can it get bigger? Well, what we need to remember is what we're recording. We're recording the activity of thousands of neurons. So what is happening is not that any action potentials are getting bigger, but cells that previously weren't firing action potentials now are firing action potentials. I said before that EEG signals are very difficult to interpret because you don't know where they're from. So the positive and negative fields often, the positive and negative waveforms don't really mean anything. But what we can look at is the frequency components that the EEG is made up of, and we've probably heard about alpha waves and beta waves and delta waves, and we still don't truly know, probably with the exception of the delta wave, what any of those truly mean, how they're generated. There are some very strong theories about how the gamma oscillation is generated. Alpha is not very clear at all, but you don't need to know how it's generated to see whether something is going on. So here is just some raw EEG recordings from mice that are either exposed to normal CO2 levels or low CO2 levels. And you can see that the frequency components that make up their EEG change. So the hypocapnic animals have more power in the low frequency range and less power in the high frequency range, which typically would infer that they were losing some kind of consciousness or losing arousal. So, more complex analysis are very common. I talked about this in 311, right? So this, for instance, is the map of the cortex made solely by looking at signal coherence, which is to say the EEG is very similar looking. And you can see that this matches very closely to some of the classical cortical regions, S1, M1, there's A1, there's V1, premotor regions. These will be the ventral stream areas down here. Point is, because the EEG signal itself is kind of a bit hard to interpret, you need to look at more complicated measures to try and draw something from it. Single unit data, as I already said, has been used to record place cells. That's one of its most famous examples, and this is the first recording of it here, where there's a rat freely exploring the environment, and every time the rat goes to area A, this neuron fires heavily. It seems to fire a little bit in point B, but really has no interest in firing in E, F, and G. So you can see that here's the raw waveform. Well, this is after it's been filtered, so each spike is an action potential. It's worth pointing out this would not pass muster now, right? This is plainly not single unit activity. There's at least one neuron here and some other neurons here. But this is supposed to be the behavior when the animal's in area A, and then here is the behavior when the area is in animal D. The animal is in area D. And you can see what they've done is they've binned the data over time. So instead of looking at the raw waveform, they're just saying how many action potentials are happening every second. Here's another incredibly famous experiment by Wolfram Schultz looking at how dopamine neurons fire in response to reward. Now, what we have here is each row is a single trial. Each little dash is a neuron firing, and then everything has been aligned to when the reward was delivered. And the point here is that with no prediction, there's no sound, just randomly the rat gets a reward. You can see these dopaminergic cells fire, 200 milliseconds after the reward. Now, after the rat has been trained, here is the conditioned stimulus, which is just a little beep informing the rat that it's going to get a reward. You can see the neurons fire in response to the conditioned stimulus, but do not fire in response to the expected reward. And then in this example, the poor rat is given the beep to tell it it's about to get the reward, but instead the reward is omitted. And you can see that the dopaminergic cells stop firing. Now, really, the point here is to get you used to looking at these peri-stimulus time histograms. So we have a histogram of the data and it's peri-stimulus, so it's around the stimulus. A PSTH is a very popular way of displaying neuroscience single unit data. But it's also just worth going into what this experiment revealed, right? That there's been this idea for a very long time that dopamine cells are rewarding cells, and when dopamine cells fire, you feel good, and if they don't fire, you feel bad, or something along these lines. Now, what Wolfram Schultz went on to show, and I think pretty convincingly, that what they really show is some kind of reward prediction or the reward prediction error. The point here is that when this rat is untrained, if a reward pops out of nowhere, the rat had no idea that it was about to get a reward. It had no idea that something good was about to happen. So the brain wants to figure out, what did I do to make that reward happen? And that is the point of the dopaminergic signal. Now, I can't convince you about that bit, but note what happens, that when here's the reward, here's where the good thing happened, but the dopamine cells don't fire. The dopamine cells fire in response to the conditioned stimulus. The stimulus, which in and of itself is non-rewarding, right? There's nothing pleasant about a little beep. What's even more interesting, and it's not shown in this figure, is if you train the rat that before the beep, there's going to be a light flash, well, the dopaminergic cells will start firing in response to the light flash, right? So what's happening is that the rat is learning what is predicting the reward, and it's trying to move its prediction earlier and earlier in time.